All right, today we're going to be talking about the second part of chapter 12, or the last part, 12.3, where we're going to look at DNA and how it's replicated. That'll lead us right into our RNA chapter and how we make proteins. So I simplified what's in your textbook up here. I have over here, I did a quick little DNA molecule, much like the one that hopefully some of you have showed me and sent me pictures. I definitely do want to see this. Okay, it is something that you can do. It's pretty easy. You can always go to the school and pick up the hard copy. If you're ever in Hardin on Monday, Tuesday, or Wednesdays, and pick up hard copies. I make hard copies for all my students. So they should be there. You can get those like from 12 o'clock to 1 o'clock whenever they're doing the lunch handouts. So we have our DNA strand. It's a double helix. Okay, it looks like a spiral staircase, the one that I have in my classroom. We've unwound it, showing our chromosomes. We know that there's a base pairing rule. A always binds to T, G always binds to C. And I also mentioned in the last video that when A binds to T, it's always a double hydrogen bond. When G binds to C, it's a triple hydrogen bond. And it's a good way of identifying which base is which base of the four letters that we need to know. But it's also to remember that hydrogen bonds are the weakest of the bonds that we've talked about before. And that's what gives this simple molecule a lot of flexibility, because we can break those bonds. And if it's easy to break them, it also means it's easy to form them. So in this last section, we get over onto this one here, and we talk about how do we make copies of our DNA. How in the S phase of the cell cycle, when the cell's going to make copies so that it can divide and form more cells, how do we make copies of all that information, that genetic information? Well, we have a bunch of enzymes that are going to be playing a role, okay? The one that your textbook talks a lot about is polymerase, DNA polymerase. And what's nice is that this polymerase, it's signified by DNA. We're going to talk about an RNA one too. One that is not in your textbook, and I don't know why they don't put these in your textbook, is called the helicase enzyme, okay? And the helicase enzyme, its job is to unzip those hydrogen bonds. It's like Velcro. It like breaks the Velcro bond, those little hooks and loops. And that's what a helicase does. And you can get multiple helicases and multiple polymerases working on a single DNA molecule. You can't think of these being done one at a time 46 times. There are multiple enzymes doing this at the same time, and then they put them back together like a big Lego set, and they do a really good job. Okay, so as this unwinds, and I just did one big loop here, I put my letters on here, made it look more complicated than it is. Okay, on this right side, the polymerases read to the top. On the left side, they read down, if you want to really get nitpicky about how the polymerases read each side, and they build the new copy. So we have our original, and then we have our new copy. This is going to give us two copies of the same information. Okay? So the polymerase reads up, reads down. The helicases unzip it. The polymerases zip it back together. And they start and stop at certain sets of letters that tell them to do that. So when this one started, it started down here. When this one started, it started right here. And it will end when it hits another set of letters. And we're going to talk about those in a second. So again, it's a way better picture in your textbook than I have here. This is just my little quick down and dirty sort of one. We have our original down here. And who knows, maybe there's another helicase that hooks on down here. And it starts unzipping this little section here. Okay? So these helicases unzip. The polymerases put them back together. And these polymerases are so good at putting these back together and putting all these little sections back together, they make one mistake every billion times. Okay? One mistake. That's like in a billion. That's you starting your whole high school career. Let's say your whole career in school and only missing one question on one test. That's how good they do. Okay? So that leads us to these letters. We know from before when we learned about them that we have the 4 nitrogen bases. I'm just going to call them 4 N bases. Okay, these 4 nitrogen bases follow the base pairing rule. A binds to T, G always binds to C. Okay, and then we know that these 4 bases, okay, are going to also be used in sets of 3 called triplets or codons. Okay, codons are sets of 3 letters. A, G, G would be a codon. C, T, A would be a codon. A would be a codon. So we have these 4 bases, but we use them in sets of 3. So this was the start codon right